All right. I think we can start. Am I audible enough?
Yes.
Okay.
Audible
Thank you.
enough.
Yes, you are, Brian.
Thank you. Thank you so much. And welcome, colleagues, to our 8th laboratory virtual CME, a series that we started to create the culture of learning within the health field, especially on the laboratory, touching into all related topics in our area of practice, and then which will help us gain more knowledge and also help us with accumulating the number of CPD points we need for the competency and also for our license renewal with the EMDC. So today we are honored to tackle another interesting topic, antimicrobial resistance. I think you remember last year, around November, December, we had 2 series which were introducing us into just general microbiology. And then part of that series is also an aspect on antimicrobial resistance. So today we have Mr. Admire T. Makunde, who is an expert in microbiology, working as the medical laboratory scientist within the AMR project in the laboratory. So without any further ado, I'd like to introduce him, and then Mr. Makunde, over to you. You can share your screen and start your session today. Thank you.
Thank you, Bangani, and hello, everyone. I think I'm audible enough. Let me try to share my screen. Is it clear enough? Let me just switch to. Is it clear enough now?
Yes, it's clear. Thank you.
Yes, it's clear.
Okay. Thank you very much for giving me this time. As Bongani has indicated, I'm Makunde Atmaya, a medical laboratory scientist by profession. So I will go through what's called antimicrobial resistance, just an overview. We won't be in a position to just more go into detail on the specifics, but I'll just give an introduction on what antimicrobial resistance is. It's just a build-up, as Bongani has indicated, that from what Andisco has shared on the testing methods which are available. So it's an addition on how antimicrobial testing done, but we need to understand how the resistance is coming about. So I will just start by a general definitions on what are the what's AMR, which is antimicrobial resistance, then the global crisis as a result of AMR, and then who are the drivers, or technically I would say, who can we say are the causes or the people who are involved in spreading the AMR. And then for us to understand more the AMR, we need to understand the mechanism which are used by big bacteria to acquire those resistance. Then as a laboratory people, what are our roles in AMR response? Do we have a part to play? Do you need to be in only microbiology for you to be considered as a player in AMR? So I think by today we'll be in a position to answer those. And what's the role of the laboratory in prevention and control of AMR in our facilities and in our communities? So let me just quickly define what antimicrobial resistance is. Usually it will be now and then you'll be hearing AMR. Antimicrobials, let's just start by defining that. Those are medications or medicines which are used to prevent or treat infectious diseases in human, in animals, in plants. I have intentionally included animals and plants, and I think at the end of the presentation, I think we will need more like to say answer why I've included the animal and the plants when we're talking about antimicrobes. So when we're talking about antimicrobes, we'll be referring to the antibacterial which usually called antibiotics, but I think I will give a broader definition on what's the difference between antibiotics and some antibacterial. Antimicrobial also referred to antiviral, also refers to antifungal, and also refers to antiparasitic. So these are the infectious organisms which you can encounter in life. So when we're talking about AMR now, we're referring to any of these organisms when they have evolved or with the mechanisms that they develop to protect themselves from those antimicrobials. By evolve, it means at one point in time it was susceptible, but because of subjected to other pressure, it end up or more like having a mechanism to survive in those pressure or hard conditions. So for you to define AMR, it's more like initially it was susceptible. Then as time goes, it was now, it's now totally resistance. So when we're talking about resistance again, there's relative and there's complete. By relative is, I think when we're talking about our antiseptics, we classify them as bactericidal, bacteriostatic. So it's the same principle on antimicrobials to say there are some which are which totally kills the organism. That's complete susceptibility. So when an organism is fully resistant, to such we say complete resistance. When it's partial, of which we need to redefine what's partial, it's not like it just inhibit, like the bacteria possesses certain characters which doesn't, like, it's not fully resistant, but it's partial resistant. It can grow, but the growth is slowed. I think we'll give an example on how we can demonstrate that in the laboratory. So we have said when we're talking about antibacterial, it can also be called antibiotics, but I need to just more like extend the definition to define the antibiotic and why it's commonly, like why antibacterial always referred as antibiotics. So antibacterial is a broader term that refers to agencies that inhibits or kills the growth of bacteria. It includes antibiotics and also synthetic antibacterial. So in antimicrobial, we have got synthetic and also we have antibiotics. So the antibiotics are those antibacterial that are naturally produced by organism. It can be fungal in case of penicillin, also streptomycin. Those are typical examples of antibiotics to say that driven from other living organism, and they've got the potential or they inhibit the growth of other microorganisms. Then synthetic, those are man-made. There's no natural production, like, so those are chemicals which are produced by human and don't have natural origin. We're talking about ciprofloxacin. Those are man-made. So I think the difference is more clear. So technically we can say all antibiotics are antibacterial, but not all antibacterials are antibiotics. But for the sake, because it's a common term which will be always used, but now and then as laboratory people, we need to understand the difference. So I will refer antibiotics in this presentation as like antibacterial or antibacterial is antibiotics, but it's just for a disclaimer. And also in this presentation, antimicrobial resistance will be more focusedly on bacteria. Remember, we said antimicrobial resistance is not only focused on bacteria, but antifungal, viral, also parasitic. Even the broad extension will now be separating to say, also talking about EMR in TB differently from bacteria. So why is AMR is the concern, or why is it being talked about more and more? It's at a global crisis that and it's not recognized, but the impact is very severe, and it won't be noticed, especially in areas where there's limited resources or data, where there's no testing, or where there's no follow-up. Let's say, what's the postmortem of death in a patient? There's no that follow-up, and we'll never hear that that was a result of AMR. Normally, we hear that it's just as a result of pneumonia, but we don't know whether it was bacteria or it was viral or other factors. So almost 700,000 people is WHO, like 2019, have been dying of drug-resistant infections. The number will be higher if you talk about bacterial infection. This is the only number referring to those which are resistant, and it's predicted that in 2050, the number could rise to 20 million deaths per year, and it will be surpassing death caused by all cancer types. It can be breast, prostate combined. So that's why there's more talk on the AMR. Antimicrobial resistance, just a snapshot, it can be a natural process that happens through genetic changes in the bacteria, and it can also be acquired, like from other resistance organism. So we are saying it's a natural process which happens over time. So if we're talking about over time, should we get worried or not? We need to be worried on those bacteria which are acquiring resistance from others. You may have a bacteria which have acquired resistance over 100 years, but if it grows within another organism, it can share that resistance mechanism just within 1 hour of growing together. So that's why it's a major concern. And why is it a concern? Is because the spreading is accelerated by human activities, like the practices which we do, which involves misuse, overuse of antibiotics in treatment. It can be human treatment, it can be animal use. Some antibiotics, I think we understand, that are used as growth promoters in horticulture, like in chicken farming, it can be pigs. Like also in animals, antibiotics are misused or overused in controlling diseases or infections within farming and also in plant growth. So human beings are the drivers of acquired resistance. And as I said, this can happen within the shortest period of time. Under normal circumstances, if everything is stable, natural process can take, and it takes a longer period, but the accelerators, which are mainly human practices, which leads to acquiring of resistance from other pathogens. I'll try to explain later on what really happens for you to end up resistance. So I'll just try to share a pictorial representation of what really happens when we're talking about resistance. By definition, we said initially the organism was susceptible. So if you expose it to antibiotics, it must die, as indicated by the first picture. But due to resistance, those organisms will resist or will keep on growing within an organism. Just imagine, let's say organisms are mixed. You've got one organism which is resistant, the other one which is susceptible. If you use antibiotics or if overuse antibiotics, those which are susceptible will die, those which are resistant won't die, and you are promoting them to increase in number, thereby easily spread within the community. So I'm just introducing to you to say how is overuse end up promoting resistance, or end up we have so much resistance within our community or within our health facilities. The next picture is just trying to demonstrate how the laboratory can demonstrate resistance within bacteria. So the first plate, is more like a similar plate, like organism, which was grown some years back. Then the next plate is now an organism years later. So you can see that initially it was susceptible to most antibiotics. By a clear zone, it means the organism is destroyed by the antibiotic on the white disc. Then by no clear zone, that's when we can say a resistance. So typically that's how it's demonstrated within the laboratory. So just to overview, to say what are the roles of the laboratory in AMR prevention or fight against AMR. So you have to, like, the laboratory plays a big role in performing susceptibility or resistance testing to guide rational use of antibiotics. You are the one who says it's working or it's not working through certain tests which you do within the laboratory. So it justifies whether the clinician select a certain antibiotics over the other. I think when we're initiating someone on antibiotics, the goal is total elimination, not partial elimination, of which the laboratory guides in total elimination. Where there's resistance or where there's partial resistance, it's partial elimination, and it promotes resistance or spreading or those organisms to other patients. Let's say it's within the clinical setup. The laboratory also contributes to a national and regional treatment guidelines. Like, I think all along, or all we have been reading in the text, there are guidelines where you just say, if you've got a diarrhea or if you've got a fever, if you're coughing, these are a set of antibiotics which you need to take. So the question will come, who came up with such a guideline, of which as a nation or regionally, we need as a region, we need to come up with those treatment guidelines. So it's based on the data which is generated within the community. One thing which we need to understand is resistance pattern is community-based because it circulates within the community. So what happens in region 1 shouldn't be used to what's happening in region 2, because it's highlighted the resistance, they were never acquired in the same place. So it must be treated as a separate case. That's where the laboratory plays a role to say each facility has to generate an antimicrobial resistant data, which can be used to come up with the guidelines. Guidelines, they can be at a facility level, they can be a national level, they can be at a regional level. But for you to decide on that, statistics has to be generated on resistance patterns. Then the laboratory also plays a big role in detection or alerts of outbreaks of resistance pathogens. We'll give an example of TB. For you to say there's XDR, it's a laboratory test. It's not by focusing on the patient, but it's on laboratory tests. So those are the importance of the laboratory and how it contributes to fight AMR. By generating those data, you are triggering control mechanisms which can be put in place to combat the spread of those resistant bugs. Then the end result is more like you contribute data to the global AMR surveillance, so that scientists will end up developing more or newer effective antibiotics, and also resting or more like focusing on things, on development of rapid kits which can detect certain resistance, which can be picked within the shortest period of time. We'll use an example of TB because more like data is readily available, like the gene expects are rapid, they can easily detect XDR, which initially before it was taking weeks to generate such data. So rapid tests you can only motivate them once you've got data which pinpoint certain resistance within a certain bug. So I was saying we'll cover on who are the drivers. So for an organism to end resistance, what really happens and like who are the possible causes of those or such resistance? Like mostly it's overuse or misuse of antimicrobials. So the overuse and misuse, it comes in many ways. So by just treating an organism with certain antimicrobial without knowing its resistance, there are some organisms which are naturally resistant to certain antibiotics. So let's say you take amoxicillin, you treat a coffee or a sore throat. There are some organisms which grows within that area which are naturally resistant. It's also a mixture of other organisms which are also susceptible. So what you are doing is you are killing those organisms which are susceptible, then promoting the growth of those organisms which are resistant. I'll give an example to say, let's say you've got Staphylococcus aureus, a basic organism, which is within the blood. You've got fever, but within the same blood, you also have got Klebsiella pneumoniae, which is also in circulation. If you are just using or without laboratory, that's misuse. By misuse is without a guiding information. So you treat that with amoxicillin. It only targets the Staphylococcus, but Klebsiella is naturally resistant to amoxicillin. So you are promoting the growth of Klebsiella. By that promoting, you are allowing it to spread. It can be to other organisms, to other individuals, or to other sites where it was not infectious. So by misuse, you can contribute to resistance. How is promoting overgrowth of a resistant pathogen while destroying the susceptible organism? I think you understand that naturally there's uniformity or there's no overgrowth of organisms because they're sharing the nutrients. So by that, they're suppressing them, causing infections. But if there's something which is overgrown, there is a high likely emergence of infection. One quick good example is in case of Candida, when you take antibiotics, usually that's when it starts showing symptoms in urinary tract infection. It has destroyed other bacteria which were competing with nutrients with the Candida, but now they're now suppressed. Candida will overgrow. Candida is resistant to the antibiotics. Then also misuse or use of antibiotics in other industries other than the healthcare, like we're talking about animals, we're talking about within water treatment, or even disposing. One will ask, where would be the water which is disposed within the hospital or from the pharmacist with a chemical or medicine producing companies? Where is the water going? Is it not the same water which we are going to drink? In that water, are there no antibiotics? Then that antibiotic which is excreted, is it high enough to cause total destruction in the water, or it's promoting the resistance within within the antibiotics? So overuse within health industry or other industry like farming. It can also be industrial, like where they are producing either detergents or any antimicrobial substances. It's a big driver of AMR. Then over prescription, and inappropriate prescription. By over-prescription is giving one or like more than one antibiotics to treat an organism. We understand that there are some scenarios where you need to be so sure. Let's say a patient is after a theater, like then there's an open wound. You don't want that patient to be more like infected at all costs. So you end up be giving prophylaxis. It can be more than two or more than two antibiotics. So by that, you are promoting, as I said, to say some organism dies, some are resistant by nature. So you end up more like accelerating the growth of those resistant organism over non-resistance. And organism, like I think we understand by nature, we learn to live within what's provided within the environment. So organism, bacteria, they also do that. To say once you expose them to a lower concentration, which doesn't kill them, with time, they try to learn, or that's mutation, they'll try to mutate to live within those conditions. Thereby, that's a process of acquiring resistance. Then one of the major driver of AMR, I think all of us were culprits. We never finish the course of antibiotics when we feel better. And also, there's also an assumption to say this medication has worked for me. So let me share with a colleague or a family member, which is also like having the same feeling. So you end up not finishing the course by leaving few remnants of antibiotics, and those not and bacteria, thereby they end up being learning how to resist. So all courses of antibiotics, it has to be completed. If it's not completed, it's promoting resistance. There's a common knowledge which we normally hear when we're having antibiotics, when we're visiting the pharmacy, because we won't be knowing the organism which is causing the infection. We just have a fever, we just have a sore throat, we just have a wound which doesn't resist to any treatment. So usually the first choice of antibiotics, if you don't have laboratory results, that the broad spectrum antibiotics. Typical example is amoxiclav. I think is something which you always get in pharmacy. It targets majority of the organisms. There are the good things about it, but there are also bad things about it. AMR solves everything, but it does the job. You can clear some, but you are also missing some. So broad spectrum antibiotics contribute more to resistance. Remember, I said eliminating some and leaving those which are resistant. Then there's also a narrower spectrum. So this is the role where the laboratory is coming. So the laboratory is trying to be pinpointing without affecting other organisms, but only focusing on the organism which is causing resistance within, which is resistant to certain antibiotics. So narrow spectrum antibiotics are the best choice when you are treating or when antibiotics are administered. It doesn't have effect on the normal flora. It doesn't have effect on the other organisms, which are more which are not isolated from the site where the infection is, of which that's the role of the laboratory to say, you come in here to provide that narrower antibiotic, which would be more useful and more effective without disturbing the normal flora within the system or the ecology. So this is how resistance is spread. Like we're talking about animal, we're talking about plants, we're talking about human beings. So antibiotics are used no matter where. It can be within animal production, it can be in human, but the end result is resistance, which is going to affect the human beings, which will be within the hospital. In animals, there's overuse of promotion of growth, and usually also as a prophylaxis. Like at a certain time, I think we understand those who have been keeping chicken, you have to give them certain antibiotics. And the tricky part is most of the feeds which you give, those commercially prepared feeds, they usually have those antibiotics, even which are silent, like they won't state. It's more like that. Say it's their secret, but that's the issue. Some people say, I want those traditional chicken, which have never yet been fed with animal feeds. Or it's also applicable in cattle, like also in pig. There's that usage of antibiotics. Then what happens to say some organism within those animals that spread within the community? It can be through water. It can be the ones who are taking care of those animals spreading to other human beings, or it can be also transferred within the meat, which will be eaten. Within the meat, there will be residual antibiotics. By residual antibiotics, is those few minute antibiotics, of which those are major promoters because they're in lower concentration of. So they easily promote AMR when consumed by human beings. They get into the circulation, not effective on organisms, but the organisms that are learning, the bacteria are learning to say, if you are exposed to this chemical, which is a sort of animal, you have to learn how to survive within it. Then the human beings also the same, misuse where there's a flu or any other health-related cases without knowing the resistance pattern, you start taking the antibiotics. Also, some antiviral that are treated with bacteria, some bacteria that are treated with antiviral. So that alone without lab information will promote resistance. So that's why I was more like I intentionally include animals and plants. Say everyone is involved, or in almost every sectors that are involved in promotion of resistance. So it's more like from this, it's also on how to prevent the resistance. To say action should be taken in all the sectors. It can be animal, it can be human health, it can be also plants. They need to play a role in prevention of AMR. So how do these bacteria end up resistance? There are several mechanisms where the bacteria become resistant to antibacterials. So we're talking about enzymatic inhibition when the bacteria produces an enzyme which inactivates the antibiotic. This happens over time. One typical example is resistance of penicillins. It has to penicillins, the bacteria have evolved in such a way it produces a beta-lactamase enzyme which cleaves the active part of the benzene, the penicillin rings. So I found that some organisms already have it. Like I'll give an example of Klebsiella E. coli. Almost 90% of them are all isolates at current moment. They now have those enzymes which inhibits or beta-lactam containing antibiotics. Then there's also another mechanism where there's alteration of bacterial membrane. By alteration, I think for antibiotic to get through the bacteria, there are some protein membrane which it needs to bind to for it to have access. By principle, membranes or cell membranes that are very selective for something, a large molecule to get in or inside the cytoplasm, it has to be transferred with the transfer proteins or porins, of which some bacteria, they evolve or mutate in those proteins, inhibiting the entry of antibiotic, thereby rendering its effectiveness. So it's also one mechanism where it alter. Again, on the penicillin, it's also a typical example, like the benzyl penicillin, how most organisms like Staphylococcus is resistant, is just stop entering of benzyl penicillin. It can also be oxacillin entry into the bacteria. I think usually we'll be hearing as methicillin resistance. The principle is on the alteration of the membrane. Then another mechanism is rapid injection of the drug, which is usually referred as efflux. The antibiotic gets into the cell, but before it exerts its activities, the pumping mechanism which expel or eject the antibiotics outside the cell, so they end up not being active. This method of or mode of resistance is usually exhibited by Pseudomonas, also exhibited by Acinetobacter, and is mediated by usually by gene like transfer or acquired resistance within as a result of phage, like phage which will be transferred from one organism to the other. The trick part of this is in the laboratory, you can do your 24-hour AST, which is antimicrobial susceptibility testing. You find out it's susceptible. But then when the doctor starts initiating the patient, because this organism can grow within those with the plasmids, it can be shed, then it can start resisting. You test the same organism, a day later, it's now resistance. So it poses a challenge of which those are like a common mechanism, which is very instant, which doesn't happen over a long period, but it can happen even within hours, where there's sharing of genetic information within organism. The other mechanism or how the bacteria can evolve or alter the effectiveness of antibiotics is by altering the targeted sites. It can be in ribosomal targets. It can be enzymes which are involved in metabolism. One typical of resistance which happens is only on cotrimoxazole, like when there's disturbances within the metabolism within the there's a change in metabolism within the bacteria, thereby rendering cotrimoxazole ineffective when administered to the bacteria. So this is just a summary or a pictorial picture on how the organisms can be resistant, also with some examples or common examples on certain antibiotics, how the organism learn how to resist them. When we're talking about resistance, I think I'll be more detailed to say there's some which are natural, there's some which are acquired. By natural, an organism is naturally the antibiotics has never worked. Then acquired is through mutation, and that mutation can be any of these four different mechanisms. Of note, when we're talking about the resistance, let's say we're talking about the penicillins. I have said there's binding on the cell membrane. There's also inactivation of the beta-lactam ring. So when an organism can be resistant to same type of antibiotics using different mechanisms. It can be having a multiple mechanism to say it inhibits the cell wall, and also it might have the influx pump. That's when we're talking about MTR. So an organism can have multiple modes of resistance. It can be to same antibiotic or several antibiotics. So you can have modified on the cell wall, you can have also antibiotic inactivation in the same organisms. So what's our role as a laboratory people? As I indicated earlier, when I was just giving an overview to say is to guide patient management and reduce overuse of antibiotics by providing the actual working antibiotics, which are narrower and which has been demonstrated within the laboratory. The laboratory also provide information and screening of resistance within the hospital to prevent infection and also plays a role in control. And now and then the laboratory is supposed to provide tests of environmental swab to the hospital just to have so that the hospital can improve the cleaning method. You mustn't find certain organism within the hospital environment. So the lab provide a role in infection prevention so that control measures can be put in place within the hospital. Also, the laboratory provides AMR surveillance for monitoring resistance trends. The laboratory can produce several data. I think we are used, or commonly as a laboratory, we are used to just generate lab results. The doctor will ask, where are my results? Then they generate. Then when we're asked to do a monthly statistics, we're just more like how many tests were done, 20 urine, 30 stool, then we're done. But when we're talking about surveillance, it's more like analysis of the trends of susceptibility and also resistance. It can be within specimen type, within also bacterial type, but those are statistical analysis which give an overview or insight to really to say which are the organisms which are circulating, of which that's the role of the laboratory. And that information, again, also can be used in prevention of infection, like the IPC. It can also be used in coming up with the treatment guidelines within the hospital and also nationally. Also, AMR surveillance plays a role in the global world where you provide information to researchers and also people who can assist in tapping the AMR. So those are some of the roles which the laboratory can play. So how do we screen for resistance? Usually the test which is done is antimicrobial susceptibility testing, which is usually called AST, but it's more ideal to use antimicrobial susceptibility and resistance because you are checking whether it's susceptible or also resistance. So AST, it will be referring to the testing which is done whether to assess one organism, is it resistant or is it susceptible? So it's done in vitro, like you do it outside the body to assess certain antibiotics to predict whether they are effective in vivo or not. And so technically it's used to guide the doctor to say, if they use this antibiotics, is it going to work or not? You have to demonstrate that it works in the laboratory for it also to work in the human beings. Tricky part is not this, like once you get susceptible to say certain organism is susceptible, it doesn't mean it's going to work in a patient all the time. There are other factors to say infection site, it may differ. Some antibiotics are absorbed better to the infected site than the others. So an organism, let's say E. coli, can be isolated from CSF. Then one antibiotic can work on E. coli in the CSF or in causing meningitis. But because of drug activity, some drugs, they don't cross the meninges for it to be effective. So when you are providing the antibiotic or AST results, also pay attention to the specimen source to say, is it too effective to certain, is it too absorbed to certain tissues? So AST is performed per bacterial strain from those individuals which are presumed infected. This part is more like the laboratory plays a role in deciding whether a certain infection is a normal flora and it's not a normal flora. We don't usually do AST on normal flora, but I'll explain later, like the criteria on when to do the AST. So we said AST idea is to demonstrate whether one organism is susceptible or resistant. And I emphasized on you must pay attention on the sample site, where is the sample is collected. Then also the other part which you need to pay more particular attention is the clinical data or clinical information which the doctor provides. Those are some of the things which will guide you whether something is a normal flora or something is infectious organism which have been isolated. How is it done? Most AST is done by growth method. So you have to grow an organism. Then from that growth, that's when you subject it to antibiotics. But the other methods where you don't need to grow the organism, like I think recently there's too much talk on PCR. We can utilize PCR and not even grow the organism on the culture basis. So you don't need those incubations, which you need overnight. Then the second day, where you do a pure culture, you can just go to PCR, but there's a catch with PCR to say, does it provide susceptibility or does it provide resistance pattern only? So mostly the commonly used and the best ones, which would give you a decision, is growth method. I'll outline different methods which would be used based on the growth. So you subject an organism to an antibiotic. That antibiotic, it can be on a disc or it can be in liquid form. How you do it is you have to have a pure culture of an organism. By pure culture, I think in the laboratory there's a common term which is saying isolation. By isolation, you are trying to separate all possible organisms within a specimen to come up with pure culture. So once you have that pure culture, that's when you can do AST. I've said there are several AST methods which are used. Of note, these AST that are guided, like the steps that are guided by certain standards. I'll go more on the standards as we go, and we now more like the specifics. So the commonly used standards which are used when you are doing AST is the CLSI guideline and UCAST. These are updated now and then, like almost on a yearly basis, or when there's a major change within the resistance pattern, or if there's new information on AST or resistance pattern within a certain organisms. There is just, there is this sharing of information which happens in the laboratory when we're talking about quality controls. In chemistry, in hematology, and in other departments, usually we say controls, they need to be done on a daily basis or before you start your day or when final results or the results which are not getting coming out. In bacteriology, there's usually less talk on controls. But I want to emphasize to say control, like quality control procedures are very critical in microbiology. It has to be done routinely to check on the antibiotics which are using for testing to say, are they still performing well? There are a lot of factors which can interfere with antibiotics, from storage, expiry, and also different batch numbers. So these controls, they need to be run. You also need to check on your medias, like whether they're performing well. Some antibiotics are pH dependent. So if your media, when you're preparing it, it changes the pH, they are going to be, the results are going to be affected. So controls, it's a routine thing which is done in microbiology, just to check your reagents, also your consumables. This can be done using also like a control strains, like those strains with the known or predicted outcomes. So the methods which are used for AMR, they can be classified as those phenotypic, as I said, demonstrated by culture. Phenotypic testing is, or a method like from genetics, is phenotypic, is something which can be expressed or which you can be seen. So when we are saying AST or AMR is phenotypic, it's something which you find in the laboratory to say, I've grown this organism, did it grow or it doesn't grow? So it's demonstrated by culture on a plate to expose the bacteria to antibiotic, then we check whether it's resistant or not. Then there's a genotype method, which is PCR. It only checks the resistance, so the targeted genes or resistance, which you'll be checking. Remember, I said AMR is as a result of mutation. So wherever there's mutation, this can be demonstrated using molecular method. So it's something which you don't see with your naked eye, but it's something which can be demonstrated when you do PCR. There are other biochemical methods which are now in use, like you can use chromogenic media. You can use also rapid kits which are available. And usually those, again, like the genotype, they usually tells you to say present of resistance and not telling you susceptibility pattern. So the advantages of each versus to the other. But phenotypic is usually the one which guides and routinely done. Genotypic is usually done for surveillance to say is the spread of certain bacteria, and also for knowledge to understand really the type of mechanism, like the type of genes which causes the resistance, but not to guide on treatment or not to check whether treatment has responded well or not. So what's the duration of getting the AST results? Like when you're using the phenotype method, which is culturing, which is more like considered as the traditional method, which is still the method which is in use to guide treatment and also change of therapy. Usually it takes 48 hours. First, you have to culture the sample to check whether there's growth. Then you have to subculture and do AST. So probably the results will come the 30th day and the 40th day, depending on whether you get a pure colony or not. So basically the turnaround time, usually when you get it, is more like on that is 3 to 4 days. There are also some organisms which are slow growers. So you might end up getting the pure culture, maybe after 40 days of culture. That's why the turnaround is a little bit prolonged. But there are other methods which have shortened. They are still culture-based, but they made shortening the subculture period, which is the broth method, which can be automated. I'll go into it to say how can it be shortened and how can you shorten it from 40 hours to almost 24 or not to almost 36 hours, or get after subculturing, you can get AST results within 10 hours. Then also indicated to say that some selective media, so they reduce the AST period by just subculturing and end up just picking the resistance as early as possible. But remember, we said the disadvantage is it's just resistance only. It doesn't share the susceptibility antibiotics. It can be PCR also, which we can generate results within 45 minutes or 2 hours, but it's only on resistance. There are also some rapid kits where it's similar to the ones which you are using, the way you just drop a suspension, then you wait for almost 2 hours to check whether there's any resistance gene within the cultured bacteria. So we said culture-based is the ideal way. So it can be done using the broad AST method, where you suspend the pure culture in tubes, or it can be micro, like those ELISA tubes, those micro titer plates, where you do serial dilution. The serial dilution is done on the antibiotic. Then you put suspension of pure colony into that. So that serial dilution, it will guide you whether at a certain concentration, the antimicrobe is effective or not. So when you're interpreting broad methods, the last concentration where there's no growth, that's the concentration which you use. On how you interpret it, I think I will cover more on interpretation of MIC and also disk diffusion. Then also the second method which can be used is antimicrobial gradient diffusion method, usually it's referred as e-test. So different concentration of antibiotics, of one antibiotic is put on a test strip, like it's just a strip with a different concentration. Then your culture is similar to disk methods, but the difference is the strip has got different concentration. I will show you how you interpret that. Then also the third method is the disk diffusion method. I think we are more familiar with this method. The round disk impregnated with antibiotic or microbes, then with a specific concentration, then it's put on a pure culture and incubated over time. Then zone of inhibition is measured depending on the standards. Some is 18 hours, some is 24 hours of growth. That's when you can interpret the results. So the disk diffusion, if I indicated, usually the most standardized way, remember we're talking about when you are doing AST, there are some standards which you follow or guidelines which you follow. And these guidelines are indicated to say they are usually updated on a yearly basis. So those are the ones which you need to follow. So the most standardized method in disk diffusion is the Muller into an ACA, which is usually referred as Power method, useful in most of the bacteria or bacterial types, but it's not useful in some bacteria. Like we can talk about Neisseria gonorrhea, it won't grow on Muller endon agar, it grows on certain specific. We talk about Streptococcus or Haemophilus influenza, it doesn't grow on Muller endon agar. So, but it's a standardized method for most organisms. There are other methods which can be used in those organisms, which I have indicated. The commonly used within the region which we are is the CLSI guideline. And it has got like the procedure on how to do AST on different organisms and how you can interpret whether resistance or susceptible per antibiotic disc. So those antibiotics, which are those white discs, they have got a known concentration of antibiotics which is impregnated So they're dried, like they're commercially prepared. So what you do, you prepare suspension and stick on the plate, then you put those discs, you incubate over a period of like 24 hours, but it depends on certain antibiotics and certain organisms. I'm specific on that because some organisms, some antibiotics, it's 18 hours. Those are standardized method. Some antibiotics that 24 hours, like I'll give an example for all interpretation of vancomycin, disc AST, it's a must. You have to read exactly 24 hours after incubation. Other antibiotics, it's 16 to 18 hours. So the guideline will explain which duration which you need to do. So the same disc method can also be used other than just checking on the AST. It can be used to check other phenotypic resistance. Remember, I said there are several mechanisms of resistance which you can encounter. This picture, like on your, like where it's like a D, like which is below, you can use the disc method to demonstrate inducible clindamycin resistance. This is a phenomenon which is exhibited by the Staphylococcus and also Streptococcus species, to say they might be resistant to clindamycin if they were exposed to erythromycin at some point in time within the circulation. Then there's also another phenomenon which can also be demonstrated within using the disc method, like the ESBL, like the extended spectrum beta-lactamase resistance, which is usually in the E. coli Klebsiella group, like resistant to the cephalosporins, which can be demonstrated within the lab. Like usually you put so this arrangement is very critical when you are doing AST, like we are saying. The first one, which I said, clindamycin and erythromycin, those are discs which are put together. In the laboratory, you don't need to say day 2, you are just randomly putting the antibiotics. Then day 3, you are trying to determine the phenomenon of resistance. So it's best way to say when you are placing the antibiotic for OST, you must know which discs to put together. So the first case, erythromycin is supposed to be put closer to clindamycin when you are testing the gram positives. Then when you are testing the gram-negative bacilli, like the third generation cephalosporins, must be put closer to amoxiclav or any clavulinic acid to demonstrate if there are any like enzymes which hydrolyzes. Like if you can check between the picture, the plate which has got those 4 discs, you can see some areas or between the other, like the one in the middle, there's that resistance, like there's that more like notice it's not like a uniform inhibition zone. It's a mechanism of resistance which can be demonstrated using the disc method. So the other methods which can be used, like I indicated to say, there's also an MIC. I think you can see the strip which is written gradient disc diffusion. It's the same as the disc method, but the difference is the strip will be having different concentration as indicated, like those numbers. They indicate the concentration, different concentration within the strip region. So when you expose that strip to an organism, where there's a higher concentration, there's bigger inhibition zone. Where there's smaller concentration, there's a smaller inhibition. Where there's total or an absence of inhibition, that's where you said is the MIC. By MIC, you are saying it's the minimum concentration which is effective to kill the organism. That can take 18 hours. So the turnaround time of the E-test, which is a gradient test, is 18 hours. from setting up the AST. Then there's also the micro plate, which is the tube broth method, where you have to do different concentration. It takes almost 18 hours, but with improvement, it can take 6 hours. This method can be automated within the laboratory, the micro plate method. It can be automated within the laboratory and make it even more faster, like 6 to 8 hours. For those fast growing organisms, we are talking about E. coli. It's a fast grower, so you can get AST results. Like you incubate from specimen culture, it's 24 hours. Then from 24 hours, you just add 6 hours. So it's almost within 30 hours from sample collection. You might have results, but it's not organism which gives such the turnaround time. Is only for those fast growers, as I indicated. So on the interpretation, if I earlier on highlighted, there are some things which you need to say. Some AST that are not done or interpreted without the clinical information from the clinician. Like we're talking of commensals, like Staphylococcus epidermidis. Usually it's isolated in specimen like blood, joints, and CSF, but as a result of contamination. But you won't be knowing this because the same Staphylococcus epidermidis can cause blood infection in cases where the patient is immune compromised or open wounds, like those bigger wounds. So you need the clinical information for you to interpret or to release and also do AST on certain bacteria. So the other consideration which you need to pay attention is to understand the existence of natural susceptibility and also understand also natural resistance, which is intrinsic resistance. By natural susceptibility, there are some organisms by default that are usually susceptible. I think we have heard of biochemical tests which we're using Optocin on strep pneumo and also novobiocin. It's one of those test antibiotics where a non-susceptibility, natural susceptibility to say an organism in any way, it will never acquire any resistance. So that those non-organisms versus AST. Then intrinsic resistance, those are organisms which at any point were never susceptible. They have a natural gene within or a mechanism within them. Typical example is E. coli is naturally resistant, and also most gram-negatives that are naturally resistant to vancomycin. Not that they have a gene, but it's just vancomycin is a larger molecule antibiotic which can pass through the cell membrane or those proteins which I have highlighted within, like to say they are the ones which promote absorption. So naturally, most gram-negatives, they don't take in vancomycin. So it's naturally resistance. There are several natural resistance which you need to pay attention. And in some labs, they are used as biochemical markers to say, in as much as you know resistance exists, but you also need to know whether your identification is correct or not. Klebsiella is naturally resistant to ampicillin and amoxicillin due to the presence of the beta-lactamase enzyme, like the specific gene is the SHV1. So that's where you need a sequencing or PCR to demonstrate the presence of certain genes which promote resistance. Then also one of the common, to consider, let's say protease is naturally resistant to tetracycline and colectin. It has got a natural target which reduces binding of those antibiotics. There are the list of antibiotics, of which I think this picture is misplaced, where I've indicated, or I will share, I think it will come out, where there's a list of organisms which are naturally resistant to certain antibiotics. The guideline which you'll be using usually provides those lists. When you are interpreting results, also you need to pay attention in clinical breaking points. You usually need to say reference point, which will be used for you to say resistant, susceptible, or intermediate. You need what's called breaking points, depending on the methods which you are using. If you are using DISC method, the breaking points is the diameter of the inhibition zone. If you are using MIC, you need MIC, the smallest concentration or the minimum concentration where the antibiotic was susceptible, or when it starts exhibiting certain resistance. So for you to come up with the breaking points, certain comes in, and it's very practical. And also there are some statistical tools which will be used. You have to there are some tests which will be done to say, a subject is given the antibiotics, you check the mechanism of action, whether it's absorbed. So you're talking about pharmacokinetics and dynamics, whether that organism is released to the tissue. Then also check the concentration within the blood. Then you also check the clinical outcome. So the clinical outcome to say one person was infected after giving this antibiotic at a certain concentration, were they responsive? By responsive was the infecting organism disappear or killed? That's when that minimum concentration or the smallest concentration of antibiotics given to the patient, and it's effective at that particular point. That's when you define MIC. It's difficult to do on a routine basis. That's why you use international guidelines where reference methods are used or to come up with those concentration, because you have to measure the actual concentration of antibiotic within circulation, and that's when you can come up with decisions. And this MIC or the breaking points, they change depending on the available information. Breaking points for one organism might differ also because of the site of infection. So you can have breaking points for E. coli, which infects the lungs, different from E. coli, which infect in UTI, just because of the pharmacokinetics and dynamics. To say the bladder might inhibit certain antibiotics, so the concentration is less than the same antibiotic given to someone with pneumonia. The exposure will be high at a specific time. That's why they have those breaking points. So breaking points are interpretation values which you use, whether to say this is susceptible or it's resistance. So from this MIC, that's when you can also get the inhibition zone, which is used for DISC method. The MIC, you get it when you are doing the gradient test, which is E-test. You can also get it when you are using the micro dilution, which is the broth method. And this method is easily automated. Most available equipments which can do AST, they use this method. One typical example is the Vitek 2, which uses the broth method. Then the zone breaking points is driven from the MIC. So you need to have an MIC for a specific antibiotic. Then that's when you can determine the inhibition, the DISC inhibition zone. it also involves you do test at a larger scale, then you come up with a statistical analysis, not done again at a facility level. That's why you have to use guidelines. So for you to interpret, you need results, whether it's susceptible or resistance. You need clinical breaking points, which are provided by the guidelines, which you are following. So there are several guidelines, as I've earlier indicated, depending on the lab specifications. Usually, the outcome is the same, but how you interpret the results and how you do the tests might differ. So these standards, these are documents that is developed through a process of consensus. By consensus, is different labs participate in checking the activity of certain antibiotic at a certain concentration. Then there's an agreement which you need to come up with. It was effective in different settings. Then they also outline the material which you need for you to do AST. So different standards uses different material, different method. I'll give an example of the commonly used one, the clinical, the CLSI guideline, and UCAST. One of the major difference, all of them, they use molar indon acre, but the difference comes on the CLSI uses sheep blood, then UCAST, it uses horse blood. So it's more like on the material which you need. Then also some on the breaking point, on the concentration of antibiotics. CLSI usually it uses higher concentration of antibiotics on disc, and the UCAST it uses less concentration on the disc. But the good thing, there are efforts to harmonize those standards into one standard for global use. Other countries or other areas, they use the British Society Anti-Microbial Guide. All these are not like mandatory for a certain, like the lab is not asked to choose to say, like it's not given the standard, say use this one, the lab decides on which guideline, but you have to indicate it in your, to say, I'm using this standard just for interpretation reasons. So in the laboratory or in microbiology, these standards are usually considered as the Bible of AST, or the normal Bible which you use to say, there's nothing which you can do or which can decide without following what these guidelines are. From how you do the test and how you interpret the results. Once you have chosen your Bible, you cannot change things to suit you, or you have to change completely. Like it's more like you're changing religion completely. You don't mix those things. You can use it on several platforms to say you are using automated, you are using this guideline, you are using this method, you are using a different method. You must use a standardized, and it must be shared with the hospital setting where you are sending the results, because for them to interpret the outcome, it has to be from the guidelines. As I like on the selection of antibiotics, what I indicated earlier on to say it depends on the bacteria isolated, whether it's natural or acquired. So that's the first thing to say. The organism which you have, does it exhibit certain natural resistance? So basically what it means is the lab plays a bigger role in basic identification. I think that era of saying gram negative and you release the results, I think if you understand natural resistance, you can see that it's not really ideal. You have to be specified to say it's E. coli, Klebsiella, or Proteus, or Staphylococcus aureus, because there are implications on selection of antibiotics. You might just gram negative, then I go on the gram negative list of antibiotics, I select those ones, but a certain, like let's say Klebsiella might have certain resistance, or Proteus, if I have indicated to say tetracycline is natural resistant, all E. coli and proteus, they all cause UTI. They're all gram negatives. So if you report gram negatives, if I give tetracycline without knowing the actual species of the organism, it might also have an implication. Then also how you select or how you test the antibiotics, it's based on the methods which are used. Some methods, they don't allow this method. Some, like I'll give an example. When you're doing strep pneumonia, some MIC, because of the complexity of how it grows, it might not favor the MIC method, but also favors the disc method. There are some scenarios where disc methods don't work. Example is vancomycin, usually on staphylococcus, it doesn't have breaking points, so it's only MIC which is acceptable. So the test method also determines the selection of antibiotic selection. As I indicated earlier on, sometimes test methods are just done to check whether identification is correct or whether identification is not correct. So it can be used as a biochemical marker, indicated to say ampicillin or amoxicillin can be used to confirm whether really it's Klebsiella cases. Then just for information, sometimes when you are choosing antibiotics, there's a term which is usually referred as antibiotic equivalence. By antibiotic equivalence, to say in the laboratory you have the ability to test certain antibiotics, but they're in the same family. I talk about ampicillin versus amoxicillin. They're in the same like penicillin group, but amoxicillin, because it's not stable to be kept in the S-disc method. Usually the best method to demonstrate its susceptibility or resistance is to use ampicillin. So once you get ampicillin results, it's as good as we can also recommend like amoxicillin results. Another example is in the laboratory, it's easier to test for erythromycin than to test for clarithromycin, which is commonly dispensed in pharmacy. So once you get erythromycin results, you can predict the outcome. So it's usually considered as antibiotic equivalence. This information needs to be shared with doctors because we have seen some cases or there are some scenarios where clinicians will say in the certain laboratory, you're not doing this antibiotic, but this antibiotic exists in our pharmacy, of which you are doing it or the laboratory is doing it, but the issue is they're not sharing like those antibiotics which they are saying antibiotic equivalency. Natural resistance, as I have indicated, I'm sorry, it's just this presentation is just a mix-up. This is the table which indicates some of the organism with natural resistance to a certain antibiotic. The ones which are indicated as blue, those are resistance or natural intrinsically resistance to certain antibiotics. Penicillin G, E. coli, Klebsiella, Enterobacter, and Serratia, and Pseudomonas. All these organisms, they are naturally resistant. You don't need to test it. Then ampicillin and amoxicillin, Klebsiella, Serratia, and Pseudomonas, natural resistance. Then the selected first, second, third generation cephalosporins, where Pseudomonas, Enterobacter, and also some gram-positive like Enterococcus, where there's natural resistance. So you don't need to test this organism on those antibiotics on those organisms. Then, as a laboratory, we are there to provide accurate and timely antimicrobial results that to assist in prevention and controlling of infection. We must also provide information and surveillance and outbreak. Remember, I said there's a difference between just issuing results and do participate in surveillance. In surveillance, you have to analyze or monitor the trends which like microorganism exhibit within a facility. That will assist in preventions on potential outbreak. The laboratory also plays a role in controlling AMR by collaborating with the infection control within the hospital. That will assist them to clean up. You can do swabs in theater, you can do swab with the ICU, neonatal wounds, bench unit. That will assist the cleaning or a change in the antiseptics, which can be used for treating certain antibiotics. Also, the laboratory can play a bigger role in antimicrobial steward program. These are where almost all departments within the hospital have a representative in just coming on analyzing and how to prevent antimicrobial resistance by coming up with guidelines which can be used at a facility. The laboratory can play a role in research, like on evaluation of new testing methods, those rapid methods, whether they're effective and non-effective. So accurate data can be shared so that informed or quicker decision can be decided on those resistant organisms. Just as a take-home message, I think I've tried to share like what AMR is and what we can do. So the take-home message, you can take it as a laboratory person and also as someone who lives within the community. We need to understand that the effectiveness of antibiotics is running out and there's less development. So we need to prevent it in one way or the other. AMR is not discriminatory. Like the impact is the same within the health. And also it's it impacts the economy. There are certain, like it's very expensive to deal with infection. I think we've seen it in COVID with some resistance. Like a lot of money was channeled to that. We know of TB, we know about HIV resistance. There are resistance which can be avoided, of which it's also seen in other bacteria. To say there is that overuse of money. So it's better to prevent than to just wait and see. We have got a role to play as a health professional and policymakers and also in the society. We can advise people and we can also play a role in avoiding misuse of antibiotics and also provide information, accurate information to the clinicians on treatment of the patients. Then there's we need to collaborate. Remember, I said the laboratory plays a role in infection prevention. The lab has to participate, scientists have to participate in those activities within the facilities. It can be infection prevention control, it can be the stewardship committee. And then we mustn't wait for others to act. It's everyone's requirement to put effort. I thank you. Then this is one of the useful links, like where you can get more information, like some in short courses, some just videos, which would be demonstrating AMR. I've just to share some of you. So thank you. And I appreciate ICAP and EHLS for most of the information which I've shared. It's from them and experience which I've gained from them. Thank you.
Well, thank you so much, Mr. Makunde, for such an enlightening and comprehensive presentation on AMR. I must say that it's quite interesting to learn such information in terms of AMR, which is a very big part of microbiology that it's gaining momentum, And as lab professionals, it's the right space for us to equip ourselves in terms of mastering the techniques and also the knowledge you need to get in terms of AMR. And the issue of lab testing accuracy, timely reporting, and adherence to the established guideline cannot be overstated, as they are paramount in terms of informing the clinical decisions. And colleagues, I must also allude to the fact that we also have a lot of learning material in terms of AMR, which are CPD accredited. We will share, I think, latest next week, which are also a build up to this presentation by Mr. Makonde. And at this point, we'll just take a couple of comments, maybe some reactions from the participants. I'll start with the comment section. We have some comments and some question. Maybe let me start with Mr. Konde, commenting, saying we hope that now we have numbers, which is the data on the widespread of AMR in Eswatini. And then another question from him, asking that are there any measures being put in place to reduce AMR in Eswatini? Mr. Makonde, maybe you could respond to that one.
Yeah, sorry. Yeah, the measures which are put in place as a country, like there are a lot of activities, like there are certain like hospitals which are actively involved in the AMR surveillance studies. Like I think the EHLS is in a position to share, or Bongani share with more in detail, like what's happening in terms of the AMR surveillance, but the country is actively involved. Thank you.
All right. Thank you so much, Mr. Makonde. Yeah, maybe just to add, I think we do know that the AMR project has spread to all the mother labs or the regional labs in all the regions. And also we have the Sentinel sites, which are working hand in hand with the clinics to ensure that all samples are tested and they go through the AMR project, which is part of the project by ICAP and EHLS to monitor the resistance pattern in the country. Still on the comment section, there's another one maybe from Zile Zedlamini asking, okay, maybe let me read the whole comment. Thank you for the insightful AMR series, and thanks, Mr. Makonde, for the excellent presentation. Given the substantial drug resistance, we've identified our laboratory testing existing. Are there existing platforms, initiatives where we could collaborate with therapeutic committees and other relevant stakeholders to discuss AMR data and inform drug procurement decision? I'm asking this because we think collaborating would enable us to make more informed choices based on this specific resistant pattern identified in our country procuring antibiotics. Yeah, I think in a nutshell, maybe before I give to Mr. Makonde, it still talks on the big AMR project, Maybe would you like to add something, Mr. Makonde?
Yeah, thank you, Bongani. It's almost similar to the previous question, but just to add on the platforms and also participation on collaboration. It's advisable whether the hospital or the facility is doing bacterial testing. It's advisable to come up with the AMS committee, those antimicrobial steward committees. Already the facilities are using antibiotics, so why not, even if there's no laboratory, play a role in just control the use of those antibiotics. It also promotes like lab tests, as Bongani indicated, to say facilities can refer to the testing lab. I think that is open, like EHLS, I think it's open to that. Thank you.
All right. Thank you so much. Still on the comments, I think most of the participants, Katlego, Mtiko, Talsile, appreciating and Mobile appreciating the well presentation from you, Mr. Makonde. Thank you so much, colleagues. And I'm not sure, before we close out, anyone that would like to raise a hand, maybe ask a question or a comment. Okay, I think in the interest of time, colleagues, just like we usually do, as we close, I think we are all used to quality that if it's not documented, it's not done. So just to test the knowledge retention of this wonderful presentation, in the chat box, I've posted a small quiz which acts as an attendance register, colleagues. Don't mind the score that you'll get, but it's just another platform for you to test how well did you understand what was shared here today, and maybe what would prompt you to read further in terms of your research in terms of AMR. So I've posted a link on the chat section. And as we close, I would like to appreciate you all, colleagues, in tuning in this CME series, learning a lot. And this session is recorded, I must say. Like I mentioned last time, we have a YouTube channel where we post all these recordings and the quizzes, which is like an attendance register for you to mark that you've participated or learned. So it's also in the description section. So please take your time outside this platform and also continue learning. And we'll still do share a lot of online short courses which are approved by the CPD program and also recognized and also help you in your continual learning platform. So from me to you, colleagues, thank you so much. As we close, please take a moment and complete the quiz. I will also share my screen here so that you can scan the QR code and do the quiz, colleagues. Thank you so much.